the recording to start? Okay. All right. So thank you, Francois. So I'll begin my talk. So as Francois said, I'm a professor at the Université du Québec in Montreal in the computer science department. And I'm also the director of the LINUM, the Laboratory of Digital Imaging, Neurophotonics, and Microscopy that I funded in 2020 when I arrived at UQAM. So my talk today will be about imaging ex vivo whole mouse brains with neurophotonics to improve tractography algorithms. Just to begin, I'll explain the motivation behind my work and what I will try to explain today. So basically, the main goal of the research we do in my lab is to better understand the fiber crossings across the brain to improve diffusion MRI tractography algorithm performances. And in that goal, what we want to do is to go in specific areas in the brain and image different fiber crossing topologies like bending, fanning, crossing fibers, kissing fibers, and different other kinds of geometry. And the goal is to be able to know at a very fine scale what is happening in the fiber bundles in those locations, to then try to use that information as a priori information to better guide the decision the tractography algorithm makes in those areas. So the question we can ask is, how can we describe the local tissue organization or the mesoscale structural connectivity with neurophotonics tool? So so that's the main goal of my presentation. So today I will first introduce what neurophotonics is, and I will try to focus on all brain imaging of fiber orientation and structural connectivity. I won't cover everything. I will mainly focus on the main techniques as a quick introduction. Then I will explain the specific microscope we use in my lab. So this is serial OCT, serial optical current tomography. And finally, I will show a few slides on example of how we can use serial OCT to infer the fiber orientation in the goal of later on study fiber crossings at micrometer scale. So that's basically the plan of my presentation. Okay, so first, what is neurophotonics? We can use two simple definitions. One comes from Wikipedia and the other one from the Neurophotonics Society here in Quebec, in Quebec, Canada. So the first one is basically neurophotonics is the use of microscopy or spectroscopic methods in neuroscience. So that's really wide. Anything that uses light to study the brain is basically neurophotonics. Another definition from the Neurophotonics Society is harnessing the power of light to study the nervous system and understand the mysteries of the brain. There are a lot of neurophotonics technique, and here is just a summary, and I didn't cover everything. So we can use neurophotonics, for example, to do functional imaging with fNIRS. So with fNIRS, we use near-infrared light with captors we put on the head, and then we can measure infer the brain activity by the change in the blood in the blood at the surface of the brain. We can also map the neuronal activity if we do calcium imaging. So with that, we can see individual neuron light up and follow what is happening at the circuit level. We can also do optogenetics. So if we modify, if we use transgenic mice, we can basically use a laser to activate some parts of the brain. So there are different ways of doing this. We can combine acoustic method with optical methods to do functional optoacoustic. So with that, we can, for example, map the vasculature. We can do nanoscopic imaging, synaptic nanosurgery. So there are a lot of techniques, but in my lab, we focus mainly on 3D histology using optics. So why do we want to use neurophotonics? As I said before, our goal is to study white matter architecture and structural connectivity at micron scale. So that's the main keyword here, in all brains using optics. So if we want to better understand what is happening in individual crossing fibers areas, we need to go at a very high resolution. Currently, in our lab, we work mainly with mouse brains, but we also can work with rat brains. And in the future, we also want to adapt the techniques I'll present today to macaque brains and parts of human brains. Why is neurophotonics interesting for our applications? So if we compare what we usually use to study the brain and to do tractography, so diffusion MRI, here is an example of what I did a few years ago during my PhD. So for the same brains, we did diffusion MRI with our high angular resolution sequence, the RD sequence, at 125 micron per voxel. So these were acquired with ex vivo mouse brain. So for those brains, what I'm showing here is a template of B0 template for the brains we imaged during my PhD. So every voxel here is isotropic at 125 micron. For the same experiments, we also acquired the same brains in 3D with what we call the OCM, so optical coherence microscope. And we were able to register all our OCM images and find where they are in the MRI brain. And just to compare, the OCM, the lateral resolution in the image plane is 1.5 micron. So we can see a lot more detail about the fiber architecture. So here, what I want you to realize is with microscopy, if we are able to co-register the microscopy data on the MRI, it gives us a whole other view of what is happening in the brain. The field of view, the size of the image of the OCM is the same as a single MRI voxel. So it gives us a lot of information that we can then use to try to improve the tractography algorithm to use that as a priori information. So that's the main motivation behind using neurophotonics in my lab. I will present two ways of doing optics to study the brain. The first one is what I call targeted optical imaging. So with this technique, we use different labels or contrast agents or different dyes to mark specific structures in the brain or different molecules in the tissue. So that's typically what we will see in histology, for example, with H&E stains. Here, I will show that in a few slides. We can also do immunohistology to target specific proteins in the brain. And on the right, this is an example of a previous experiments we did where we injected FITC, fluorescent protein, inside the vessels during the sacrifice of the mice with a gelatin. And then that revealed that made the whole vasculature fluorescent. So with our system, our serial OCT, our serial 2-photon system, we're able to map the whole vasculature in 3D in the whole mouse brain. So with targeted imaging, we have various techniques to do imaging and to label the tissue. So the one that is the most frequent is histology. So just to quickly summarize how histology works, we have a sample, we take a small part of the sample, a biopsy, or there are different ways of taking this. Then we have to fix the sample to prevent it from deteriorating during imaging. So this is typically done with PFA, formalin, there are different ways of fixing the tissue. After that, we embed the tissue in a medium. So in our case, you will see later, we use agarose, but you can also use paraffin or epoxy, depending on which type of histology we'll do. Once the sample is embedded in a block, you section it, you cut it into thin slices. These slices, the thickness will depend on the kind of microscopy you will make. And once you have the slices, you can apply different stains or different staining techniques to reveal different parts or different structure in the brain. So typically what we will see is H&E staining, and we'll image that with optics. But we can also use electron-dense stain to do electron microscopy. We can do histochemical staining, to reveal different parts. We can reveal where some enzymes are present in the sample. And we can also do immunohistochemistry, where we use an antigen that will link to different proteins, and then we label this antigen or we functionalize it with the fluorescent protein. So if we emit some light, it will emit another wavelength, and then we can map that in the sample. Another thing I want you to realize is with histology, the appearance of what you will see will depend greatly on how you will slice the sample. So for example, if you slice it you do cross-section, if you have a vessel, for example, you will see the same fibers here will appear completely different as if you do longitudinal section, or you can also do sections in other orientations. So we need to keep that in mind when we do neurophotonics, histology, or these techniques. What we will see will depend on our point of view or on the way we sliced the sample. Okay, so histology is a way of preparing the sample, and then to do the imaging, we have different techniques. So one of those techniques is fluorescence microscopy, where we will use a dye that will link to different parts of the sample, and then we will illuminate the specimen with an excitation wavelength. So, for example, we can excite it with a blue wavelength, and then the dye will interact with it and re-emit a different color. So for example, here in the example, the color is green. Then we use a filter to remove the excitation wavelength, and we measure the remaining light, the fluorescence light. So that's a really frequent technique, and fluorescence microscopy is often combined with what we call confocal microscopy. So instead of imaging with the whole specimen without filtering anything, so for example, with a camera, with confocal microscopy, what we do is we add a small hole between the detector and the sample, and we also have a hole between the light source and the sample. So by adding this light, what this will do is it will limit where the light will come from, where the fluorescence light will come from. So all the light can only come from the focal plane. All the other light that could emit fluorescence further away from the focal plane will be cut by the aperture. So the advantage of this is it will remove all the light that cause blurry images. So for example, if we don't have the apertures, this is what we call wide field microscopy, and the sample will appear as really blurry. So I can zoom on that part. This is an example of the blurry images if we don't use the pinhole. But if we have the pinhole, this is what we call confocal microscopy, and the image becomes a lot less blurry. We can see more finer details. With confocal microscopy, we have to often perform a scan. So we will scan the laser on top of the sample, and we'll measure the light the fluorescence at each spot, and then by combining this raster scan into an image, we get a full 2D view. And if we want to do a 3D acquisition, we have to with a motorized stage or with something that moves the objective, we have to change the distance between the sample and the objective, and this will acquire a different stack that we can then combine together to get a volume. So that brings me to the first application of this technique to study the tissue organization in 3D. So that's a paper from 2016 from Kurt Schilling that used basically 2D confocal microscopy, and they performed different stacks. So they did a scan at different depth inside the tissue. And from that, they then showed a processing pipeline to fix some artifacts that came from the optics, made sure that the data was So we did PSF, they did PSF deconvolution, they performed interpolation to get an isotopic resolution. And then they extracted the orientation within that volume with structure tensor analysis. So I will discuss about that a bit more later on, but structure tensor analysis is really similar to DTI analysis for the diffusion signal. And then they perform other processing to finally get a fiber orientation distribution from this data. So in that case, they use confocal microscopy. One disadvantage of the confocal microscopy is that the light, the volume where the light can come from, where the fluorescence can originate, is kind of kind of big. And also, because we use pinholes, we limit the amount of light that can come to the detector. So we have a lower SNR. We have more noise in our measurement. One way of fixing that is to use a nonlinear optical effect called two-photon fluorescence, where if we increase the light, the intensity of the light, if we have enough photon, we can have some fluorophores that will absorb two photons at the same time and re-emit the combined energy of the two photons. So that way we shift the excitation wavelength and the detection wavelength a lot more, and it's easier for us to have a filter. And also we can, because this is a nonlinear effect, the volume where this effect can happen is really small, and we can remove the pinhole. So by doing two-photon microscopy, we remove the pinhole, we have a higher SNR, and we can also go deeper because typically the excitation wavelength will be at a longer wavelength that have a better penetration inside the tissue. So using two-photon microscopy, we can also study the structural connectivity of the brain. And the example I will show you is one from this paper, the mesoscale connectome of the mouse brain, that was published in 2014. So for this experiment, what they did is they injected a viral tracer that infected some neurons in the brain. So for example, they can inject in the cortex here of the mouse. They waited a few weeks to let the virus infect the whole axon and its projection. And after that, they sacrifice the mice and they image it with a serial two-photon microscope. So I will explain exactly later on what serial two-photon microscope means, but for now, we can just assume it's a way of doing two-photon microscopy in all brains at micron scale resolution. And then they did, so this is what I just said, they did two-photon microscopy, and that revealed the whole projection map for a specific subset of neurons. And then by doing these experiments with different injection sites in different locations with over 1,000 mice, if I remember, I think it's even more. They aligned all of this in the same reference space, and they were able to create a tracer-derived tractogram. And from that, they were able to study the structural connectivity of the mouse for a population of mice. So for that technique, they use serial two-photon microscopy and not confocal microscopy. Okay. The next technique I want to discuss is light sheet microscopy. For most techniques that I described so far, well, no, I will just go back a bit. So the first technique we discussed was wide field microscopy. With wide field, we excite the whole sample, and then we measure the fluorescence from different points, all of this at the same time. So the image will be blurry. With confocal microscopy, we use pinholes, and we have to focus the light on different places in the sample. And then for each point, we measure the fluorescence. So this is a long process. We have to do a scan in 2D for every location. And then if we want to have 3D, we have to move that focal plane up and down to get a full 3D representation of the brain. To make that imaging faster, we can instead do what we light sheet microscopy. So we excite a whole slab, a whole sheet of the sample. We can do that in different ways. For example, we can use a cylindrical lens to create a flat beam. And then we measure with the camera all the fluorescence that was emitted from that plane. So instead of doing a 2D scan, we can measure everything with the camera. So that makes the imaging a lot faster. Here's an example on the bottom of what that focal plane looks like. For wide field, we image a whole sample. For confocal, we image a small beam. If we want to do a 2D image, we have to scan the beam in X, Y. And if we want to do light sheet, we excite with a sheet of light instead of doing a scan. One difficulty with light sheet is because the excitation beam has to traverse the whole sample, we need to reduce the amount of scattering in that sample to make sure that we have we keep our resolution in the light sheet. So to do this, we have to make the sample completely transparent with tissue clearing. So there are different techniques to do this. So two techniques, two examples I will show you. The first one comes from my collaborators in Bordeaux. So here they injected, again, two viral tracers, and only the neurons that were connected by both viruses after the projection emitted fluorescence. And then after that, they sacrifice the mice, they perform tissue clearing, and they image that sample with light sheet. And here, this is a video, but if you focus in some part, we can see the individual neurons, and we can also track them, see the collaterals, and we can see where those neurons project inside the brain. Instead of doing tracers, we can also use immunohistochemistry. This is an example from 2023 where they use two ways of labeling the brain, the NERN label and the myelin basic protein label. So one of them will map all the neurons, and the other one will map all the neurons that have protein that have myelin. And with that, they can also do light sheet microscopy in the whole brains and then study the architecture of the connections. Okay, so those were a few targeted optical imaging techniques where we have to label what we want to see before doing imaging. Another category of techniques is what we call intrinsic optical contrast imaging. This is where we use the existing light matter interactions in the brain to map what is happening in the sample. So we can use different techniques. We can measure what light is reflected, how much, which light is absorbed by the sample, which light is scattered. We can measure retardance, or we can use nonlinear effects like third harmonic generation or Raman scattering. And all of those techniques will give us different information about what is happening in the brain. The advantage of this is it's a lot faster to prepare a sample. So instead of waiting 1 month to make the sample transparent, we can basically sacrifice the mice in the morning and start imaging either the same day or the day after. The contrast is uniform because we don't have to worry about dye perfusion. Did we cover all the places, and it's label free. Disadvantage of intrinsic optical contrast is that it's lower. It's lower specificity. So basically, everything in the brain can reflect the light. So we have to make sure that we are able to disentangle what light was reflected by myelin and what light was reflected by other components, for example. And it's typically has a lower SNR if we compare it to fluorescence confocal microscopy. The first example I want to show is 3D PLI. So 3D PLI means 3D polarization light imaging. Polarization light imaging. Here it exploits one property of the light, its polarization, and it measures how the polarization was changed by the sample when the light traversed the slice. So here, for this technique, we basically use the fact that if we have layered structures or organized structures, they will affect polarization in different ways based on based on the orientation of polarization. I won't go into details. And with that, we are able to measure the orientation of the fibers inside the sample. This technique uses, it's a little bit more complex. They control also the tilt of the sample, and that allows them to also measure the orientation in 3D inside the slices. In my lab, we use another intrinsic optical contrast technique called optical coherence tomography. So OCT is a 3D imaging technique that can map the sample at high resolution in 3D by measuring the scattering, by using the scattering of the sample. So basically, by summarizing it, we use a broadband light source that we split into two arms. One part of the light goes to a mirror, a reference mirror that is at a fixed distance from the beam splitter. And the other part of the light goes to a sample. And the light that goes to the sample will be reflected by the different surfaces inside the sample. Once it comes back, it will interfere with the light that comes back from the mirror, and then we'll measure this interference with a spectrometer. This will give us different interference patterns for different wavelengths, and then we can reconstruct that signal by doing a Fourier transform to get the reflectivity of the sample at different depth for a single scan. With OCT, we also need to scan the beam on top of the sample. So if we do a single scan, this is what we call an A scan, that will give you the reflectivity of the sample at different depth. If we scan that beam in one direction, so basically we do multiple measurements, we get a B scan, a 2D image. And this is an example of a B scan for a retina. This is often used in ophthalmology to study the eye. And you can see the different layers inside the retina. If you scan it in X and Y, you get a 3D volume that we often call a C scan. A C scan. And if you measure other properties with OCT, for example, you can also measure how the different effect of the on polarization. You can do what we call polarization sensitive OCT, where you can then measure directly from the signal the 2D orientation of the fibers within the plane. So why use OCT for neuroimaging? There are different reasons, but mainly it's because the OCT signal is in the brain is mainly driven by reflection, and the higher reflection is in myelin because of the way the myelin sheets are organized. So without any contrast agent, with OCT, you can see myelin. So that's a really good tool to study white matter in the brain. I will accelerate because I realize I will not have enough time. So one limitation of OCT is that it has a limited penetration depth inside the tissue. So just a small example, if you shine a light on your hand, the light that will come through will be blurry. You won't see all the details of what is happening in your hand. This is due to scattering. So to be able to do 3D imaging in all samples, there are different techniques you can use. You can do tissue clearing with light sheet microscopy. You can do multiphoton microscopy, and you can do serial block phase histology. This is what we decided to use in my lab. So serial OCT is a process where we combine an OCT with a vibratome that slices a sample, and then we do an automated acquisition. We slice a sample, and then we move the sample under a microscope objective, and we acquire multiple images, and we repeat that process, slicing, imaging, until we image the whole sample. And after that, we stitch all of this data with offline 3D reconstruction algorithms. And after that, we can use that information to study what is happening in the brain. So an example of a data set we acquired recently. So this is 10x SOCT. The resolution in plane is 3 micron, and the distance between the slice in that case is 200 micron. And if we zoom in, we can see all the details of the fibers inside the brain. So here in that example, we didn't see, but we can also follow them from the cortex up to the yeah, we can follow the fibers basically. We can see it visually. We are able to track them. So that basically show us that it's probably a good tool to study the structural connectivity without using labels. So that means we can study with more brains. If we look at serial lucidity with a lower magnification, so this is 3x instead of 10x, we can look at some parts in 3D, and we can see that, for example, here in the corpus callosum, we can see the fibers going out of the corpus callosum. We also see that they are organized in small bundles that then propagate in the rest of the brain. So the OCT, serial OCT, is intrinsically a 3D technique that allows us to map the myelin in the brain, in all brains at micron scale resolution. Yeah, I won't really explain this, but basically SOCT needs a lot of neuroinformatics tools because we generate about 8 terabytes of data per acquisition per brain. So we need to automatize acquisition. We perform the reconstruction on supercomputers. We develop, in my lab, we develop a web-based tool to visualize, annotate, interact with the data. We also have to adapt the typical tools used in neuroscience to align the brains, perform different tasks. So a lot of informatics is implied by serial OCT. We have to work with big data, and that's a challenge. So some of the things we did with our system, we created a template at 25 micron from the OCT, and that's published. So you have the link here. And our template is aligned to the Allen mouse brain template, so you can know which parts of the brain correspond to what. Using that template, we were able to also map really high resolution images that were registered to our serial OCT data, and we registered that to diffusion MRI data that we perform in the same brain. And then by comparing different metrics we can infer from the diffusion signal, we saw that we had a good qualitative correspondence between, for example, FA and the structure we saw at high resolution in the brain. So these images are what we call optical coherence microscopy. The resolution is about 1 micron per pixel. And for example, for NUFO, the number of fiber orientation, if we are in the CC, in our microscopy images, we see a single direction. But if we are in a different location that add high NUFO values, we see fibers going up and down, left and right. And also with OCT, all the black spots here correspond to fibers going in plane and out of plane. So we saw a good correlation between the structure in OCT and the MRI metrics. So that basically convinced us to pursue with that technique to try to do tractography-based, microscopy-based tractography. So if we want to use the serial OCT data to perform tractography, what do we need? We need to describe the orientation of the fibers for every voxel inside our data. So how can we do this? There are different ways. I will show you 2 ways here. So the first is to use serial 2 illumination angle polarization sensitive OCT. So that's a lot of term, but basically it means that we do serial OCT, but we also measure the effect of the sample on the polarization. if we do this, I say we, but this was done with by a lab in Boston. You can analyze the signal to get to measure the birefringence of the sample, and then from that you are able to infer the 3D orientation of the sample for every voxel by basically combining the polarization with the two eliminations. And that was compared with diffusion tensor imaging, and they saw a good correlation between both. One problem with this kind of system is first, it implies to change what we have here in my lab, but it's also that it's a lot sensitive to noise. So a different approach is to perform serial OCT, but to do a population study. So we do serial OCT by slicing the brains at different angles, and then we create a template, and we can look at how the contrast of the brain structures change based on the point of view. I won't go into details, but that's basically relies on an observation a lot of people did in neurophotonics, is that in fiber bundles, the contrast change based on the orientation of the fiber in relation with the orientation of the laser. So if the fibers are aligned with the laser, they will appear as dark. If they are orthogonal to the laser, they will appear as bright. So if you control the slicing orientation, you will have high resolution information about the inclination of the fibers. And if you combine that with a method that extracts the in-plane orientation, we get a full 3D view of the orientation of the fibers. And then we can use that to do tractography in those brains. So really quickly to end, if we have images, structural images, we can extract the orientation of the structures we see in those images with a different of image, a list of image processing approaches. Some of them will compute the derivative of the image intensity. So we can use gradient approaches. We can measure this structure tensor, the Hessian matrix. We can also do multi-scale approaches. So if we go to wavelets, to directional filters, to curvelets, shearlets, those are different tools to do this. The advantage of steerable filters, and I will show you one slide on this, is that we can measure the orientation for a specific a low number of orientation, and then combine them to infer the orientation of the structure for all the other directions. So that's really computationally efficient. And another tool that combines steerable filters and multi-scale is the Ritz wavelet transform. I won't explain this today, but that's something we are exploring in our lab to analyze our data. So just a quick presentation. The structure tensor in 3D will basically compute the image gradient at every location and then combine those locally with a window function and create what we call a structure tensor. So the structure tensor in 3D is really similar to the diffusion tensor that you use in the MRI. And by analyzing the eigenvalues of this tensor at every location, we can infer the geometry of the structure we are looking at locally. So is it a fiber? Is it a plane? Is it isotropic? So it's a sphere. And then by doing this, some people show that for example, the PSOCT signal was correlated with the structure tensor results. So that shows us that this tool is a good tool at very high resolution to extract the orientation of the fibers. Instead of using the gradient, we can use higher order derivatives. So if we do this, for example, we can create a similar matrix as the structure tensor to create the Hessian matrix with the second derivative. And if we analyze the eigenvalues of this matrix, we can also infer the orientations of the fibers locally. So this is the same slice I was showing you. This is a 10x serial OCT slice where we measure the in-plane orientation of the fibers using Hessian matrix decomposition. And one last slide. This is an example of a recent work by one of our students. So he used the serial OCT attenuation template, and then he performed a steerable Gaussian quadratic filters analysis on it. and he was able to extract the 3D orientation in the brain. So if we then if we compare that, we see that in some areas we have a good correlation between what we see in the MRI and what we see from the optics, and in other parts it's not. It's a little bit less successful, but we have ways of improving this. For example, this analysis was done at a much lower resolution of 50 micron. This is really low for our kind of data. And we could adapt our technique to perform that analysis at 3 micron. So that's the kind of work we are pursuing in my lab. So to conclude, I showed today many optical microscopy methods that can use to study the white matter local orientation. And among them, what we use in my lab is serial OCT. So we use that to map the white matter in all mouse brains at high resolution without any labels. If we use advanced image processing tools, we can extract the 3D orientations of the fibers from that data. And what we intend to do with this is, first, we'll create a multi-orientation mouse brain SOCT template that will map at 3 micron scale the resolution, the orientation of the fibers inside the mouse brain. And we intend to use that to do SOCT-based tractography. So on that note, I'd like to thank you for your attention. I'd like to thank the lab members, my funding agencies, and I'm open to questions and discussions. Thank you.
Thank you very much for your great talk, great overview of everything you do in optical imaging. A lot of very long expression that you keep adding, like a new keyword at the end. It's very impressive how much work you're doing. I don't know if there is any question from the crowd. If there is any question that you would like to ask, you can unmute yourself and ask directly, or you can write it in the chat, and I will try to follow along with the chat. I don't know if there is anyone with a question. Otherwise, I can go ahead first. Okay, so one quick question for you. From my understanding, a lot of people in your field use custom build hardware. When you read a paper and you think something is very interesting, like, can you reproduce someone else's work from a paper? Do you necessarily need to contact them to ask like extra question? I'm very curious, like, how you can try to reproduce prior literature if most people have custom built hardware.
So it really depends on which techniques, but just for an example, I will use the light sheet microscope. So we do light sheet microscopy with one of my students at Polytechnique Montreal, and our microscope is based on an open source design. So the mesospin, if I remember correctly, all the parts that you can use to build that system are listed. You have the code to control it, the analysis code to stage the data, to analyze all of this. And they also share a list of labels and protocols that you can use to reproduce the experiments. So there's a big push towards open science and sharing design, sharing protocols. So for some system, we can, if we have the funding, and if we want to reproduce the study, we can build our own system. For others, you can look at the paper, and often in optics, it's all the materials is listed, even with parts number and where we bought them. So there's a way of building the system if we want. And other times also, you can buy a similar system from different manufacturers. But there's a big push towards open science and reproducibility in optics.
Thank you very much. That answered my question very well. I see that the cell has a raised hand. Can go ahead.
Yeah, it's me. Can you hear me? Yeah. Hey, Joël, thanks for the great talk. What kind of technical revolution will we need to do this on a human brain in reasonable time? Do you imagine it in your lifetime, or we need, a physics/engineering revolution that we don't envision yet to be able to do this?
Right now, I think we have there are some teams that are trying to use the same techniques to map the whole mouse, the whole brain. So the Big Brain project did that. The Human Connectome project, they try to do the same thing with PLI. I think right now the main challenge is related to the amount of data we generate. So in our case, for a mouse brain, we generate 8 terabytes, and we only measure one value per pixel for this. I think I saw in one paper, they were trying to compute how much data we will need for the whole human brain, but I think it was in the amount of petabytes for a single brain. So now we need to work on dedicated tools and neuroinformatic tools for this kind of multidimensional big data. That's, I think, in my opinion, the main challenge. And there are already some efforts to work with this. So all the community that did serial electron microscopy, they started working on data formats like the ZAR format, the OMEZAR convention for the metadata that is specialized for this. And the ZAR format can also work with Dask, so we can do distributed computing on clusters in the cloud. So in my opinion, the main limiting challenge, if we want to map a whole human brain, is the data and the neuroinformatics tool to deal with that data.
Okay, thank you.
Any other question from the crowd? You can write it also in the chat if you want. I can go ahead with another question. Twice in your talk, you mentioned that like you did some imaging, you did some reconstruction, and then it was compared with diffusion MRI, and it had good correlation. In your view, which one is like which one is the gold standard? Because it seems like you compared it with tractography, but with diffusion MRI, but ultimately diffusion MRI is an indirect measurement, so. If it did not agree with it, mean you were right and the other one is wrong, or do you see the disagreement as more indicative of something problematic with your method?
I think it depends on what you want to show, at which scale, and what interests you. So, for example, if you want to study the microcircuitry of small local networks, I think the only techniques that can give you that information is electron microscopy, where you will segment individual neurons, you will find the synapses, you will find how everything is connected together. You need that scale to study this. If you want to see long-range connections and you are interested in individual neurons, then tracer injections is probably the way to go. But you have to keep in mind that with tracer injections, you only see a subset of neurons. You don't see everything. And also, tracer injections will see unmyelinated neurons, not just myelinated. So the signal origin is different. If you want to compare with what you see in MRI, we also have to keep in mind that we measure different things. In OCT, we measure what is reflected by the myelin sheets. In MRI, you measure the water diffusion. So it's not the same thing. We always need to keep in mind that we are not looking at the same thing. So we always have to keep that, to be careful when we do comparison. So the gold standard really depends on what you're trying to show and what you're trying to improve. I know it's a vague answer, but
yeah, but I think it's an excellent answer. I knew we're not measuring something different, but I didn't realize how different they were. So that's, I think, a very good answer. Okay, so are anyone from the crowd going to ask a question? I know maybe you're shy.
I saw a question in the chat.
Oh, sorry. Yeah, I didn't get the notification.
So is OCT sensitive to photo bleaching? So the quick answer would be no, because photo bleaching mostly affects, it's when you use dyes or autofluorescence or things like this, you rely on the molecular structure of what will absorb the light and re-emit. And because with OCT, you measure just the change in index of refraction. If you don't disturb the tissue, if you don't put enough energy, you won't affect this. If you do combine imaging, for example, 2-photon with OCT, you can put too much light energy in 2-photon and start to boil the tissue. That happened once. And that will obviously change the signal you see in OCT, but that's a different effect. And the other question is, how long did it take for one slice? So for one slice, we optimize our automation code and acquisition code recently this summer. So before we were about at 3 hours per slice, and one slice is about 200 gigabytes. And now we are down to 20 to 30 minutes. And we have ways of optimizing this, but now to make it even faster, but now that would be more hardware-related work, electronics, and ways of using our computer more efficiently and transferring data. But that's a bit too detailed for, I think, the presentation. But there are ways of making it faster. And the last question: how do you compensate for the potential loss of each cut? So that's a great question. So with OCT, it's a 3D imaging technique. So we make sure that in each volume we acquire, we have an area that is common from one slice to the other. So that way we are able to stitch the data without any loss of tissue. So the quick answer is we have overlaps between tiles. Yeah.
I don't know if Kurt is still on the meeting. I see him in the description. I don't know if I don't want to put him on the spot, but if Kurt, if you're there, how do you feel about your work being cited? You know, like a couple of years later, like how do you compare both of your work? I don't know if you're here, Kurt. Maybe I'm talking to nothing.
Hi, I'm here. Hi. I mean, it's always exciting seeing your work cited, right? But yeah, when we were doing this, we had no like we were really limited with the resources and the hardware that we could use. Sorry, my voice is losing my voice. And even our 3D stacks were very small. So we did try to highlight that. And it's really cool 5 years later seeing huge 3D volumes. And like you said, not gigabytes, but orders of magnitude more data to be able to actually answer questions about the 3D structure of tissue. So I guess to answer your question, yes. And I like your question, Francois, on like how, if we see something, how we can do that at our institution or our university, because it does seem like some of these are so niche, so unique setups that it may not be possible to just say, oh, I want to buy a light sheet microscope like setup, or I want to do OCT. So I've always wondered that as well. We were limited with what we had, so it's kind of cool seeing that being used across institutions.
Thank you for answering my question, Kurt. Sorry for putting you on the spot. Any other question from the crowd? I don't see any raised hand. Let's look at the chat. Well, I think that's all. If there is no one else, I would say thank you to Joël for his great presentation. I would say thank you to everyone for coming to this presentation. Next month, we should have a presentation from Pamela Guevara for superficial white matter and tractography. It should be on the Thursday at 9 a.m. Eastern time. I will send an invite to everyone. And we will still work using the mailing list to send invitation. Maybe we'll do some trial with the IST mailing list next month. We'll see. And I hope you show up for our future webinar. Thank you very much to everyone. And thank you again to Joël for his great presentation.
Thank you.
Well, Joël, you had a great reaction in the chat, and you got 50 people, so that's great.
Ah, good.
And it was recorded. Somebody started the AI companion. I don't know what that is.
I think it's still recording.
Yeah. And do you want to stop the recording? You will receive an email notification.